Hi, my name is Haley Finkelstein, and today I'll be presenting my research project on KHDC3, a potential regulator of epigenetic inheritance. So traditional ideas about inheritance have been focused on Mendelian genetics, or the random inheritance of parental genotypes as a means of expressing phenotypes in offspring. However, recently we've begun to examine non-Mendelian forms of genetics, namely epigenetics, which is the control of gene expression, turning a gene on or off, based on the accumulation of epigenetic markings throughout a person's lifetime. So these markings can arise due to a variety of factors, such as a person's nutrition, chemical factors, lifestyle, or overall geographical environment. And specifically for pregnant women, changes to their intrauterine environment can lead to health issues and phenotypic changes due to the accumulation of epigenetic markings in the mother that are passed down to the fetus and the fetus's gametes, creating a transgenerational effect where markings accumulated by a grandparent affect their child and their grandchild. The mechanism of epigenetic inheritance is currently unknown, but small non-coding RNAs have recently emerged as potential drivers of epigenetic changes through their ability to regulate gene expression. And because of this, it has been found that changes in small RNA profiles has the potential to lead to future disorders if certain essential genes are expressed or suppressed. So epigenetic inheritance was first seen in Överkalix, Sweden, where a transgenerational response to a famine was observed, in which the grandchildren of grandparents who lived through the famine showed higher rates of obesity, cardiovascular disease, and mortality despite not living through the famine themselves. And such studies are most often replicated with mouse models. So for example, the offspring of mice who were fed a high-fat diet were shown to develop certain metabolic disorders despite not being directly exposed to that diet. And additionally, when considering epigenetic inheritance through the maternal germline, it is important to note that oocytes are very sensitive to intrauterine changes and are therefore more susceptible to accumulating epigenetic markings from the environment. So traditional studies of epigenetic inheritance have focused on small RNA profiles in sperm and the paternal germline. So this study is unique in filling a gap in knowledge by examining a potential molecular driver of epigenetic inheritance in oocytes, namely the KHDC3 gene. So this gene contains an RNA-binding KH domain that is well positioned to receive extracellular signals from its environment, and it is also expressed greatly in sperm and oocytes, so it is a great candidate for such research because it could be more susceptible to accumulating epigenetic markings, and it has also been found that women with KHDC3 mutations develop infertility and genetic defects, so it is also more specific to the maternal germline and oocytes. So this study specifically will be examining whether the presence or absence of KHDC3 in oocytes will impact the expression of two factors in the liver, lipoprotein lipase and ceruloplasmin. So the aim of this research was to evaluate the effects of KHDC3 deficiency in mice on LPL and CP expression in the liver, as well as to observe a potential effective epigenetic inheritance. So the research question was, what role does KHDC3 play in regulating liver, LPL, and CP expression? And I hypothesized that knockout mice would display differential LPL and CP expression, and additionally that expression would vary based on parental genotype, not simply offspring genotype. So 4 cohorts were used in these experiments. Firstly were a control group of wild-type mice from wild-type parents. Secondly, a group of wild-type mice from parents who were heterozygous for the KHDC3 deficiency mutation. Thirdly were a group of KHDC3 deficient mice from knockout parents whose gene had been deleted, and finally a group of KHDC3 deficient mice from heterozygous parents. And what's important to note is that the first and second and third and fourth categories respectively represent genetically identical mice who vary only in the genotypes of their parents. So this research began with the dissection of 36 livers from mice so that the liver RNA could be extracted. And then with this RNA, a master mix of buffer solutions, primers, and the RNA was prepared for PCR, a polymerase chain reaction, to produce an extended cDNA or copy DNA from our original RNA sample, with the purpose of performing quantitative PCR or qPCR. So I like to think of qPCR as a sort of photocopy machine for DNA because you're taking an original smaller sample and going through a process of reverse transcription, denaturation, annealing, and extension of that original DNA or RNA so that it is easier to view patterns and expression for your gene of interest. So with the CT, or cycle threshold values of LPL and CP expression that I obtained from qPCR, I conducted the delta-delta CT method, which is this formula you see here, resulting in the delta-delta CT value, which is a comparison of the fold change in gene expression levels with LPL and CP compared to a housekeeping gene. So initial results confirmed my original hypothesis that mice who were KHDC3 deficient displayed differential expression of LPLs compared to mice who had normal levels of KHDC3. But more interestingly, an effect of epigenetic inheritance was observed. So for wild-type mice that were genetically identical, we found that those born to heterozygous parents showed a much wider and greater range of LPL expression, and this suggests differential expression based on the parental genotype influencing offspring phenotype. I observed the same effect with the KHDC3 deficient mice, as again those born to heterozygous parents displayed a much wider and greater range of LPL expression despite being genetically identical to those born to knockout parents. So procedures were repeated with ceruloplasmin this time, and the same effect was found where within genetically identical categories of wild-type and KHDC3 deficient mice, those born to heterozygous parents observed differential expression for CP. So some key findings of this research were differential LPL and CP expression between KHDC3 deficient and non-deficient mice, and most interestingly, an effect of epigenetic inheritance was seen where LPL and CP levels varied based on the parental genotype specifically. So this research is filling a gap in knowledge by providing preliminary evidence for KHDC3 as a regulator of epigenetic markings and inheritance. So this study was limited by a relatively small sample size, as well as by the fact that mouse models are not necessarily applicable to human biology, and I only examined 2 factors in the liver here. So in the future, I would hope to repeat with a larger sample size to conduct more comprehensive statistical analysis, and I also plan to measure levels of other genes and molecules in the liver, pancreas, and pituitary for wild-type and KHDC3 deficient mice. So epigenetic research in general will hopefully someday allow us to have a greater understanding of the pathogenesis of major diseases, and also allow us to redefine disease as a product of our ancestors' life experiences rather than simply our own and our genetics. Additionally, such research could someday be used to craft health profiles for patients to predict future disease and prevent it or treat it early. And finally, epigenetics could be used in therapies to treat diseases due to the reversible nature of epigenetic markings. So overall, the aim of this research was to evaluate the role that KHDC3 played in regulating liver LPL and CP expression and to observe epigenetic inheritance through this. Thank you so much for offering me the opportunity to present my research for you today.